Hi class. Hi class. I am going to we are going to talk about the neuromuscular junction one more time real quick. So let's go back. Okay. So here we do see the action potential reach the axon terminal, and that causes the voltage gated calcium channels to open up. Calcium enters the cell. Calcium causes the synaptic vesicles to fuse with the membrane and exocytose acetylcholine into the synaptic cleft. Acetylcholine binds to specific ligand-gated sodium channels, and which causes them to be opened, and sodium rushes into the cell. As sodium comes into the cells, it obviously brings in positive charge that causes local depolarization of the membrane here. And if the depolarization is strong enough, it triggers an action potential that sweeps across the sarcolemma, runs down the T tubules, and there are these voltage sensitive DHP receptors on the T tubules, which then causes conformational change in the ryanodine receptors. And as the ryanodine receptors undergo conformational change, that causes release of calcium out of the sarcoplasmic reticulum into the sarcoplasm. Do you see that? So now we have all these calcium floating around in the sarcoplasm. What will this calcium do? Well, calcium will then go and bind to troponin. Do you remember troponin that I marked on the sarcomere? And I also mentioned you have to remember troponin because this is where calcium binds. So calcium then goes and binds to troponin, which we will see in the next slide. And as soon as calcium binds to troponin, that triggers another phase called excitation contraction, coupling phase of muscle contraction. Okay, so this is another representation. I don't want you to, this is a very busy slide and I don't want you to go through everything. I just want you to know that here, let's focus here. Calcium is released. Do you see that? So calcium comes out of the sarcoplasmic reticulum, and then calcium binds to troponin. Okay, and as calcium binds to troponin, that will trigger muscle contraction, and we will look how exactly that happens in the next slide. So here we will look at number 1 here, and this is the resting state. So we have the myosin heads, and this is the thin filament made up of actin proteins. Now the myosin head is in cocked position, and this is called the high energy state. It has notice that the myosin heads has ADP and inorganic phosphate or PI attached to it. And at this stage, the myosin molecules are ready. They're super ready to bind to the actin proteins, but they cannot. Okay, but they cannot. Now, let's say when calcium comes in, as soon as calcium comes in, calcium binds to troponin here, and that causes conformational change in the troponin-tropomyosin complex. So calcium causes change in troponin-tropomyosin complex. As a result of that, myosin can now bind to actin, as you see, and this binding or attachment of myosin heads to actin is called cross bridge formation. Okay, so now the myosin heads attach to actin and form cross bridge. All right, next, and at this point the inorganic phosphate is released, so the inorganic phosphate leaves. Okay, next the myosin heads flex. Do you see flexion of myosin heads? As the myosin heads flex, this pulls the thin filament towards the M line. So if you think of the sarcomere, know that as the myosin heads flex, that pulls the thin filaments towards the M line, resulting in shortening of the sarcomere. Okay, now this is also called the power stroke. Now for the myosin heads to actually detach the thin filaments, ATP is required. So ATP is required for detachment. As ATP binds to myosin, the myosin heads detach. And then the ATP gets hydrolyzed. As you see here, the ATP is hydrolyzed to ADP and inorganic phosphate. And due to this, and the hydrolysis powers the myosin heads to come back to its normal cocked position. So this is high energy cocked position. Then we have attachment. Then we have flexion or power stroke. Then we have detachment and going back to resting high energy state of the myosin heads. All right. Okay. So here I want you to show that this is the resting state. If you look at the right hand panel, we do see the sarcomere and we see a very relaxed sarcomere. This is when a fiber is relaxed. As you see, the myosin heads are not cross-bridged with the actin proteins of the thin filament, and the sarcomere is relaxed at this position. If you look at the bottom picture in the right hand side, this one right here, we do see that there is cross-bridge formation between the myosin heads and the actin proteins. And so there is cross-bridge formation, and the myosin heads have flexed to pull the thin filaments towards the center of the sarcomere. Therefore, the sarcomere shortens, and the Z discs, which are on either side, these are the Z discs, they come closer, resulting in shortening of the sarcomere. Okay, so since we are not in class and I'm sorry. Since we are not in class, I want you to look at this YouTube video link posted. This is a really nice one, and this will actually help you understand the neuromuscular junction and mechanism of muscle contraction a little better. Okay, we already talked about motor units, so I'm going to skip this. Okay, now the question is there a relationship between the size of the sarcomere and the force of contraction? In other words, we are actually looking at the length tension relationship. So I have drawn out 3 diagrams here. The first one in the left hand side is showing us a very short sarcomere, and this is actually showing us how things look in a sarcomere that is super short. As we see here, we see the Z discs in red. The light blue lines are the myosin or the thick filaments. So these are the thick filaments, and the dark blue lines are the thin filaments. At the end of the thick filaments, the green protein, the squiggly protein, those are titin. So the point is, when a sarcomere is short, then the myosin molecules actually run into the Z disc. So there is no sliding. Now, even if there is stimulation of the sarcomere cannot contract any further. So there is no sliding. And this results in zero tension, meaning there is no force of contraction. Okay, so myosin are running into Z disc. So here we have no sliding. And if there is no sliding, there is no tension. No tension. Another term for tension is force of contraction. But I want you to know that tension is the most appropriate word that is used here. Okay. Now the next one, or the middle one, is showing us the optimum sarcomere. And here we see that there is overlap between the thick and thin filaments, and this overlap is actually important for the myosin heads to bind to actin. And once the myosin heads bind to actin, it will pull the Z disc together. It will pull the Z disc closer, and that will result in sliding. And once you have sliding, there will be tension. So there is sliding here. Sliding. And that will lead to tension. So here, as you can see, there is optimum overlap between the thick and the thin filaments that can help in cross bridge formation and flexion of the myosin heads, pulling the Z discs closer together. Good. And then we have a very spread out sarcomere, and here we see that there is barely any overlap between the thick and the thin filaments. So there is no chance of sliding. So here also we have no sliding, and again, if there is no sliding, no force. So what is the take-home message here? The take-home message is muscles at optimum length produce optimal force because there is an optimal amount of cross-bridge formation between actin and myosin. Okay, and this really depends on the size of the sarcomere. This is the take-home message of this slide. Okay, so this is the graph, and we will focus on the graph first on the right hand side. Now at the bottom of the graph we see the decreased length of the sarcomere, then we have increased length, and then we have super relaxed sarcomere. So the x-axis is showing us the length of resting sarcomere, and on the y-axis we see the tension or the force of contraction. Here we see that maximum tension, do you see this point is maximum tension, is achieved when the muscle is between 100 and 120% of its resting length. Do you see that? Between this we have maximum tension. So if you increase the length this way, or if you decrease the length, then there is decrease in tension. So again, the take-home message is there has to be an optimum length of the muscle fiber to generate the optimum amount of force. Good? All right. All right, I will stop here and then continue in another video.